All right. Well, I think we'll get started. So, welcome everybody. Thank you for coming to the PacVec seminar series. I think this is our last spring seminar, and we have two great speakers today. So we have Maxwell Newton from San Francisco State University and Emily Calhoun from Utah State University. So I think we will get started with Maxwell's talk. Um, okay, great. And so do you prefer Max or Maxwell?
You can just refer to me as Max. Can you hear me okay?
Yep, I can hear you.
Okay,
Yeah,
perfect. Yeah,
so
Max is fine.
okay. Max is a grad student in Dr. Andrea Sway's lab studying disease ecology and is working on a master's degree and majoring in integrative biology at San Francisco State University. And his work is focusing on, I think, interventions with reservoir hosts of the Borrelia and doing some really cool, innovative new work to attempt to reduce transmission from reservoirs into the tick population. So with that, I will let you take it away, Max.
All right. Thank you so much, Jeff.
Mhm.
Go ahead and share my screen. Okay. Everyone seeing this all right?
We see your side of the presenter view, Max.
Uh, uh,
I
okay.
think you can
Let's see.
toggle.
I can do it without notes. Let me just adjust this here.
And a quick note while we're getting the presentation adjusted, I was mistaken about this being the last seminar. So the last one is on June 18th. So don't miss our last one. Apologies for the mistake.
All right, so I'll go ahead and get started. As Jeff said, my name is Maxwell Newton. I am an integrative biology master's student at San Francisco State University. I work in Dr. Andrea Sway's disease ecology lab. And as Jeff mentioned, today I'm going to be discussing my current research, which is looking at reservoir targeted treatment approaches to lower human risk of Lyme disease in the Western United States. And the first question that many people have when they hear about Lyme disease is, where does it come from and what does it do? So the majority of Lyme disease cases are caused by the bacterial pathogen Borrelia burgdorferi sensu stricto, and this is transmitted to human beings through a couple different species of ticks here in the United States, also known as vectors. And if you happen to be bit by an infected tick, this will often appear as a rash in the form of a bullseye. And some of the early symptoms of Lyme disease can include fever, chills, migraine, and even fatigue. And if left untreated, these symptoms can later develop into nerve pain, arthritis, heart palpitations, and even facial palsy. When we look at the number of vector-borne disease cases here in the United States for 2020, with the number of reported cases here on the Y or the x-axis and the disease and vector which causes it itself on the y-axis, we can see that Lyme disease accounts for the highest number of cases here in the United States when compared to all other vector-borne disease cases. And more specifically, tick-borne disease cases account for the majority of these cases as well. When we look at the Lyme disease cases reported annually here in the United States, with each of these bins representing a different case definition by the CDC, and this is updated every so many number of years to have a more sensitive description of what we qualify as a positive case of Lyme disease, with year here on the x-axis and now the number of cases on the y-axis. With the exception of 2020 and 2021, due to the COVID-19 pandemic, we can see that vector-borne disease cases are on the rise in the United States as we increase in the future. And when we look more specifically at the Western United States here in this figure, which shows all of the states belonging here in the Western US, with year on the x-axis and the number of cases on the y-axis, in the supporting table on the right, we can again see that California has the highest number of cases out of all states here in the Western US. And this is due likely to the fact that we in Northern California have a sort of hyper-endemic area for Lyme disease to occur due to habitat availability, and also the fact that living in California, it's a beautiful state, and so we have a lot of people who tend to hike on public trails and access public trails and are susceptible to a number of questing infected nymphal ticks. So even after explaining all of that, you may still be wondering about the vector itself. And to help explain that further, I'm going to discuss the life cycles of Ixodes pacificus, also known as the Western black-legged tick. And this is the primary vector for Lyme disease here in the Western US. And its life stages are made up of the egg life stage, at which point it will hatch into a larvae, where it will take its first blood meal, and then it molts into its following nymphal life stage, where it takes its second blood meal, and then finally it molts into its adult life stage, where only the female tick will take an additional blood meal before laying her egg masses in the spring. And of these individual life stages, the nymphal life stage is the most critical or most important life stage, as this life stage is the one that is most likely to transmit Lyme disease to a human being. And to help explain that a little bit further, I've included this figure here, which shows the zoonotic cycle for Ixodes pacificus. So again, as you can see, it has a 3-year life cycle. And because Ixodes pacificus ticks are obligate blood feeders, that means that they need to receive a blood meal from a host in order to survive and molt into the following life stage. So if we walk through this figure together, we can see that the larvae emerge from their egg sacs in year 1 in the spring. And as we move into summer, they begin questing for potential hosts in order to receive a blood meal. And because Lyme disease is not vertically transmitted, but rather horizontally transmitted, that means that these larval ticks are born uninfected, and in order to acquire the pathogen, they need to encounter an infected reservoir host. So in the circumstance that this larval tick in year 1 encounters an infected reservoir host, it will then acquire the pathogen, so that in the following year, when it molts into its nymphal life stage in year 2, it is then capable of transmitting that pathogen to any number of susceptible hosts. After it takes its second blood meal in the nymphal life stage, again, it will molt into its final adult life stage, where the female will take 1 more blood meal in the fall before laying her egg masses in the spring and beginning the cycle over again. And when thinking about ways that we can disrupt the interactions which are occurring here to lower the infection prevalence and density of these nymphal ticks, we often look at the reservoir hosts or the hosts themselves, as these are the actual organisms which are maintaining this pathogen in the natural environment, as well as the fact that they're much easier to treat than the ticks themselves. Of which here in the Western United States, we have a variety of host species, which range from wood rats to Peromyscus species to Western gray squirrels and even Western fence lizards, over which we have this gradient of competency. So at one end of the spectrum, for example, we may have wood rats, which are a highly competent reservoir host, which are capable of supporting a high number of ticks and transmitting that pathogen to them. Whereas at the other end of the spectrum, we may have entirely non-competent hosts, such as the Western fence lizard, which are actually borreliocidal hosts, as some of you may know, which clear the tick of that pathogen. And so this creates a sort of mosaic landscape here in the Western United States, where depending on the relative abundance of each of these reservoir host species, we can really see differences in disease risk and disease dynamics. And so this brings us to my research questions, which are again looking at topical acaricide treatments and seeing if we can target these reservoir hosts to hopefully lead to declines in questing tick burdens first, and then secondly, reduce the density of questing nymphal ticks the following year. So the way that we went about conducting this study was, again, applying this topical acaricide to different bait stations that have been modified. So we have squirrel feeders as well as these smaller rodent tubes, and these have been modified to include a fabric carpet lining on the entrance and exits of these bait stations, in which we can apply the acaricide to. And then we again bait these stations to incentivize those reservoir hosts to actually enter and come in contact with our treatment method. And this is to hopefully prevent those questing larval ticks in year 1 from being able to receive a blood meal from those reservoir hosts, so that over time, in the following year, we should hopefully see a lower density and infection prevalence within those nymphal ticks. This work was conducted at 3 different sites located in Marin and San Mateo County. At each of those sites, we placed a total of 2 plots that were spaced 500 meters apart. So this was for a total of 3 experimental plots and 3 control plots. We began our acaricide treatment in March, and this extended through May, during which time I performed biweekly maintenance on each one of these sites, ensuring that all of these individual bait stations had the appropriate amount of acaricide applied and adequate bait. Our live capture efforts were performed in April and May during peak questing nymphal activity. And in 2023, when we actually began this research, we performed live capture efforts for 2 non-consecutive nights. So we trapped once in April and once in May. Whereas this year, for 2024, we decided to conduct live capture efforts for 3 consecutive nights per plot. And this was done to try to make a more concerted effort to estimate the relative abundance of those reservoir host species that are present at our individual plots. We also performed tick drag efforts to capture those questing ticks in April and May as well during that peak activity. And we performed this twice per plot. This is a visual representation of our plot design. So each of our plots are a hectare in size, and over which we place a total of 38 of these bait stations, 36 of the tubes, and 2 of the squirrel feeders. Located within the center of this plot, highlighted in blue, is this internal half-hectare capture grid. And each one of the individual points on that grid is a trapping location. And on nights that we are capturing animals, we place a total of 2 traps per trapping location for a total of 98 per plot. And the reason for this is to establish a buffer around the areas in which we are capturing animals to ensure any animal we do capture has likely come in contact with one of our treatment stations. So over our field season, we are treating these reservoir hosts using those bait stations. We're performing tick drag efforts to collect those questing ticks. And then finally, we're performing live capture efforts on those reservoir host species in order to estimate tick burdens, as well as collect tissue samples and peripheral morphological data as well. And so getting back to our research question, our first question here is, does topical acaricide treatment lead to declines in questing tick burdens on hosts? And so this was, again, collected from all of the reservoir host species that we captured during our field seasons, and this includes data from 2023 and 2024. I should preface in saying that we just finished our field season a couple weeks ago at the end of May. So the 2024 results are still technically preliminary. We still have some work to do in terms of identifying those ticks in lab. So just keep that in mind. We performed a GLM analysis on this data, looking at the total number of attached ticks on those reservoir hosts as our response variable and comparing that to treatment type as our fixed effect, and then including other random effects such as site and species. This first figure shows the mean larval Ixodes pacificus burdens between acaricide and control plots for 2023. So we have our plot here on the x-axis and then our mean tick burdens on the y-axis. And within 2023, we immediately saw a difference between our acaricide and control plots, which was great. Our mean at our acaricide plots were 1.142 compared to 1.16. And again, performing our GLM analysis on this, we received a p-value of 2.62 to the -6. So it was great that we saw an immediate difference just in terms of the amount of burdens on those reservoir host species. And when we broke that down even further and looked at the individual species burdens themselves, here we see species on the x-axis. Nifu is the scientific name for wood rat, Pika is the California deer mouse, Pima is our eastern deer mouse, and Peter is the pinion mouse. with, again, mean tick burdens on the y-axis. We can first see that we captured a relatively similar abundance of reservoir host species between our acaricide and control plots. So for wood rats, for example, we caught 26 at our acaricide and 22 at our control. The Peromyscus species tended to vary a little bit more depending on the site, whereas certain species would be present in higher abundances at certain sites compared to others. But again, when we look at this figure and compare acaricide and control sites, we can see that for all reservoir host species, there was a significant difference in terms of the amount of tick burdens that they were carrying compared to our control sites. This data is now for 2024, so this last field season that we just wrapped up. So instead of looking specifically at Ixodes pacificus burdens, again, we still need to identify these ticks. So this just includes all ticks. So this is mean attached tick burdens on the y-axis and plot type again on the x-axis. And we again received a significant difference between our acaricide and control plots. We have slightly higher outliers this year on our control plots compared to our acaricide. But again, we see a significant difference between the two, which is, again, very reassuring in that we're receiving consistent results based off of this treatment. And then again, when we break this down by individual reservoir host species for this past year, 2024, we can see that there is, again, a similar abundance of reservoir host species that have been captured between sites and a very significant difference between the species present at our control sites versus our acaricide sites. And then when we actually look at these and compare them between 2023 and 2024, with the 2024 bars being striped here, we can see that overall we had a higher abundance of attached ticks in 2024 for most, if not all, species. And with that said, though, we still saw that there was as significant, if not more significant, of a difference in terms of tick burdens between our acaricide and control plots for 2024. And then of those reservoir host species, another interesting thing that can be gained from this plot is that the wood rat specifically, compared to all other species present at our control sites for 2023 and 2024, carried the highest overall tick burdens. And this supports many findings that the wood rat is actually one of the most competent reservoir hosts that we have here in the Western United States. Now looking at our second research question here, which is, does topical acaricide treatment reduce the density of questing nymphal ticks the following year? And this was again from the data that we collected performing tick drags in 2023 and 2024. We again performed a GLMM analysis on this data, looking at the total number of questing ticks against treatment type. And here we can see this is looking at 2023 and 2024 side by side, acaricide and control plots, and then with the questing nymphal ticks per 100 meters squared on the y-axis. And in 2023, we really didn't see any statistically significant difference between our acaricide and our control plots. But I should say that this is to be expected because, again, as I explained earlier, through the zoonotic cycle, what we're really doing is we're trying to target these larval ticks within this first year. And so we're preventing those larval ticks in this first year from being able to receive a blood meal so that hopefully in the following year, we'll see a lower density of nymphal ticks. So getting back to our figure, we didn't see anything within 2023 as to be expected, but this year we are looking at the acaricide and control plots for 2024. We can see that there is a statistically significant difference between the questing nymphal ticks between our acaricide and control plots, and received a p-value of 0.02. So it's great to see that not only are we reducing the amount of tick burdens on our reservoir host species, but we are now seeing that reflected in the total nymphal density of questing ticks at those acaricide plots. So expanding on these results a little bit, our first question here regarding tick burdens. Again, it was great to see that we saw an immediate difference between our acaricide and control plots within the first year of 2023. And moving on to 2024, it was also great to see that we had very consistent results in terms of the overall abundance of tick burdens, as well as reservoir host species captured, and then the actual response that they had to the treatment. And then looking at this data a little bit further, it's also was very interesting to note the differences in reservoir host competency and how that influenced the amount of tick burdens that were on those individual reservoir host species, as I had mentioned, with the wood rats being the most competent reservoir host that was observed at our acaricide and control plots. And then secondly, our questing nymphal ticks. It was really interesting and great to see that this is also being reflected in terms of lowering the density of those questing nymphal ticks. And moving forward, we have all of these ticks which we still need to screen for Borrelia. And so it will be very interesting and great to see if this lower questing nymphal density is reflected in a lower infection prevalence as well. So we'll hopefully be doing that work this summer and should hopefully see a similar result. And then again, comparing acaricide and control plots, it's great to see now in 2024 that we are receiving a significant difference between the number of questing nymphal ticks in acaricide and control plots. And then comparing 2023 to 2024, we did see a higher overall abundance of ticks compared to 2023. But we are seeing a significant difference between, again, our acaricide and control plots for 2024. And this maybe have been due to the amount of rainfall that we received in 2023. We received a very high amount of rainfall, and this may have allowed those tick populations, just anecdotally, may have allowed them to persist in the environment longer and led to higher abundance this year. And it's not entirely uncommon for these tick communities to vary quite significantly from year to year. But in conclusion, it's great to see that our results suggest that our treatment is successful. We're having an immediate impact on tick burdens, as well as a impact on the questing nymphal density the following year. And moving forward, it will be really interesting to do further analysis on this to look to see, again, if it's correlated with nymphal infection prevalence, as well as looking further into the influence of reservoir host competency and how diversity plays a role into disease risk. And then finally, in the future, I think it would be great to incorporate integrated tick management approaches, which combine systemic acaricides with topical acaricides to look at their contributions and when used in succession with one another. And then finally, using this work for analogous vector-borne diseases such as Borrelia mayoni, which is a much more rare, but is also known to cause Lyme disease here in the United States, as well as Borrelia miyamotoi, which is known to cause tick relapsing fever. And so with that, I would just like to acknowledge, of course, PacVec and the CDC for their support and funding, as well as our partnerships with San Mateo, Mosquito and Vector Control District, and the many California state parks and regional open spaces that we collaborate with to do this research. And then a special thanks to my advisor, Dr. Andrea Sway, as well as the many lab members and technicians who have helped with this project and made this work possible. So thank you, and I'd be happy to take any questions that you have.
Excellent. Thank you very much, Max. It's a great project. Does anybody have any questions? I had a question. So since we're also seeing a lot of Lyme disease on the East Coast and throughout the sort of northeast of North America, do you think this application, this kind of the way this is designed, would work in a different location, or do you think it would have to be tweaked to deal with the ecology of a different space?
Right. Yeah, it's a great question. I think, we have a lot of unique aspects to our Lyme disease systems here in the Western United States, as I mentioned, because of the abundance and variety of reservoir host species that we have present out in the natural environment. And so, shifting this to the Eastern United States, I think it's certainly possible to incorporate a very similar type of study and receive similar results, especially considering, they have primarily one main reservoir host species there compared to here in the West Coast. However, whether it would be practical or cost effective, I think that this is certainly a system which has been adopted by vector control agencies before and applied on a smaller scale, looking at its use along trails. And so I think that's definitely something that could be looked at further when trying to implement these sort of methods to reduce infection risk, is applying it to areas where we know that people are recreating in high traffic areas where we can have the most effect at reducing any person's risk of actually encountering an infected tick.
Yeah, that was going to be my other question, was how, if you're implementing it on a broad scale, like how do you decide, how far to go, or how, that sounds like a good strategy, targeting kind of areas where people are most likely to pick up ticks.
Right. Yeah. I mean, here in the Western United States, we have much more fragmented habitat. So it may be easier to, apply it on a larger scale in the sense of applying it over source habitat because it's much more confined, whereas in the East Coast, we have much more contiguous habitat. So it would be a lot harder, I think, to apply it kind of on a broad scale, but maybe kind of focusing, as we've talked about, more specifically on trails and areas where we know people are recreating. It would be more time, cost effective, and more practical to implement.
Great. Does anybody else have any questions? All right. Well, let's thank Max again for an excellent talk.
Thank you, guys.
Yeah. And I think we'll move to our next speaker, who is going to be Emily Calhoun.
Hello.
Hi, Emily.
Hi.
So Emily, the title of Emily's talk is Gene Flow of Larvicide Resistance in Culex pipiens Complex in Salt Lake City, Utah. So Emily is a third-year PhD student at Utah State University, and she is studying aspects of Culex pipiens, quincofasciatus complexes in Utah, and population dynamics and insecticide resistance, genetics, and movement. So with that, I will let you take it away, Emily.
Thank you. Yeah, can you hear me and see the screen okay?
Yep.
Great. Well, thank you for the introduction. I'm excited to talk to you guys about this part of my project. So the title of this project is Resistance Gene Flow in Culex pipiens Complex Populations in Salt Lake City. And so the majority of my dissertation project is looking at this Culex pipiens species complex. So Culex pipiens complex is a group of mosquitoes in the genus of Culex. They're cryptic, which means they're morphologically almost identical. They're really hard to tell apart. So Culex pipiens is known as the northern house mosquito. This is their predicted range. This is a bit old now, but this is their predicted range up in the north. And the Culex quincofasciatus is down here in the south, the southern house mosquito. And the differences between these two are specifically in climate and their ability to go through diapause. So Culex pipiens is able to perform diapause and overwinter in these colder temperatures, and Culex quincofasciatus is not. They have some differences in feeding preferences. Culex pipiens is historically known as a bird feeder, and Culex fasciatus is a bit less picky. And they have some other differences, such as like mating behaviors. Culex fasciatus can mate in closed spaces, and pipiens prefers not to, as well as some other like microhabitat differences there. And so the main concern with the pipiens complex is that they're one of the largest contributors to the West Nile virus cycle throughout the United States. So the insect vector in the middle here is the Culex mosquito. That mosquito will cycle through this transmission cycle in its favorite reservoir, which is birds. And then the accidental reservoir or hosts are things like us, humans, and horses, and things like that. And so West Nile virus is a huge issue throughout the states. And knowing that pipiens is the reservoir, it's one of the biggest concerns for mosquito abatement districts to make sure that we're targeting this mosquito. So let's get into detail about pipiens' life cycle. So the mosquito that everyone's most familiar with is this adult female mosquito specifically. So this free-flying adult will be searching for a blood meal, and that's the one that you'll see trying to feed on you or around. And it's feeding because the female is feeding because it wants blood and nutrients to make eggs. So Culex mosquitoes lay their eggs in these rafts here. It's like hundreds of eggs all stuck together at the surface of the water. These eggs will then hatch into larvae. And so these little free-floating larvae will be swimming around in the water. And this part here at the base end is their siphon. That's actually what they're breathing out of. These larvae will then sort of cocoon themselves and turn into a pupa. So those are kind of like wriggling around in the water, and you can see this head part, it's the antenna forming, there's little wings forming, it's really like cocooning itself and starting that transition into an adult, but still under the water. And then that pupa will then rise to the surface, and it will then emerge from that casing as a winged adult. And so that will start the cycle all over again. Depending on the season, if it's warm out, that can happen between like a week to 10 days. But if it's cooler out, that can take upwards of a whole month for this cycle to happen. So for my project, I'm going to be focusing on this larval stage of the mosquito. And this larval stage actually has 4 instars within that larval stage. And so I was specifically looking for this like 3rd or 4th instar, the larger stages, and I wanted to include a photo as to like what I was seeing in the microscope on the left here. You can really tell the difference. They get a lot bigger. These larvae, when you're looking at different species, it's obviously a little bit harder to tell, but you can really see how tiny that one at the top is, and then there's one hidden right here. It's so tiny, you can hardly even see it. And so as I was going through, it got easier to be able to determine like which was the 3rd and 4th instars. So larvae are a huge target for mosquito abatement districts because they're able to use what's called a larvicide, so a killing agent towards these larvae. And they're actually able to target a huge amount of larvae in their breeding habitat because they're laid in the water and they're in these like clusters. They're able to disperse a larvicide into that area and kill a large number of them. And these are well targeted for mosquito and Diptera larva itself. So it's a bit more targeted than an adulticide that would be sprayed and impact potentially more non-target organisms around the area. And so I'm specifically going to be talking about a bacterial larvicide. There's a few different types of larvicides, such as insect growth regulators and oils, but we're going to be looking at microbial larvicides. So the one in particular that I'm focusing on is called Laciniobacillus spiricus. This is a gram-positive, broad-shaped bacterium. They are actually like encased in this spore, which is also encased in a granule. So that's on the bottom here. And these granules are able to be thrown or dispensed into the water using either just like a packet or in these little granules, you can throw them out. And these granules will then sit in the water, and the mosquito larvae will actually come around. And as they're ingesting these granules, the toxin that these spores produce in this like late stage sporulation phase actually binds to midgut receptors in the mosquito larva and produces a killing agent, which kills the mosquito. There's also been, after these larvae die, there's some reports showing that it impacts like the nervous system as well as the digestive system. So there's sort of multiple things going on that are killing these larvae, but most specifically, it's these midgut receptors that end up killing the mosquito. So there has been previous insecticide resistance seen in Salt Lake City. So that was sort of the inspiration for this project, was checking years later to see what was going on within the Salt Lake City Valley. So there was insecticide resistance that was found in Culex pipiens populations in catch basins, because that is where those granules are dispensed throughout the city. And it was specifically to a product, Vectolex, and like a water dispersible granule, those pellets. And that was in 2016. It was originally noticed, and they think it was due to improper rotation of those active ingredients. Ideally, you'd have like a microbial pesticide and then a growth regulator, and you'd kind of rotate between the two because that will involve less resistance being able to spread. So they found resistance up here in the north, and so I wanted to test in these same locations to see if this resistance was still being found. Salt Lake City Mosquito Abatement District did some bioassays years later after noticing recurring resistance, and they still found some. So we wanted to do a more in-depth look in this valley to see what was going on, if there were some resistance genes that were being spread throughout the population. And so the gene we wanted to look at is called CQM1. There's some literature that shows that this bin toxin that the Licinus bacillus sphaericus produces actually that binds to the midgut and then disrupts this gene, and that's the major cause for this resistance. So we wanted to look at this link between this gene and the Culex pipiens population in Salt Lake City. And this here on the left isn't our gel, this is from the paper, but we have done some preliminary research to show that this gene is present within the pipiens population in Salt Lake. And so our main hypothesis is that populations of Culex pipiens complex mosquitoes will have differing levels of resistance to Licinus bacillus sphaericus in various habitats throughout Salt Lake City. And then we are predicting that this resistance gene, CQM1, will be spread throughout this population. And then more specifically, we want to be looking in different populations, so both below ground mosquitoes here on the right, these catch basins that are more regularly treated throughout the summer season, the mosquito season in Salt Lake, where those granules are thrown into these catch basins, and these above ground populations such as pools and ponds that are a bit less regularly treated. We want to see if there's gene flow between and amongst these habitats, and whether these Culex pipiens complex mosquitoes are sharing genes amongst them. So I wanted to use that map that I had a few slides back, the one that was published a few years ago, sort of as a guide for where I was doing my collections. And so this map on the left here shows these blue are the below ground. So these are the catch basins or storm drains, and this red up at the top are the above ground. So those ponds and pools. And I wanted to make sure that they were spaced in a way that made sense ecologically for Culex pipiens mosquitoes. So they're really only flying a few miles at most in their life cycle as they're laying their eggs. And so I wanted these above and below ground habitats to be near to each other so that the potential for gene flow was a little bit higher there, and we might be able to see some comparisons. And so I wanted these to be overlapping, and then the below ground into Salt Lake Valley in the bottom, that I just wanted to really spread down and cover the same areas that were in that paper that was previously published. So here are some examples of catch basins. In Salt Lake City, a lot of them look like these on the roads. Like you've probably passed by them so many times and never even noticed them. They're sort of just a part of the city planning. If your city is prone to flooding or something like that, this helps gather all of those storm runoff and sediment that's coming through the street. So they can look like this on the side of the roads, or these sort of like more like pothole type that are in the roads. And yeah, so you'd have to lift off that metal grate with like a crowbar or something, and then reach underneath with a dipper. So that's what I'm holding in both of these pictures. And that's what you dip into the catch basin to find those mosquito larvae. And so catch basins actually are kind of like a cool micro ecosystem in cities. It kind of provided some cool like entomology experimenting last summer while we were doing field collections. In the middle here, this is a hoverfly, and it's laying a live birth of a larva. And so their larvae are really crazy looking. They're called brat tail maggots right here. And so it really just created this like ecosystem within the city for these organisms to be like laying in sediment, but in the middle of the street, which I found pretty interesting, and is also another part of a question of mine, is that whether it's creating this like warmth underneath the ground for these mosquitoes to be overwintering in a place like Salt Lake City that gets really cold and freezes for many months of the year. Is it creating this sort of microhabitat of warmth for these mosquitoes, especially for the Culex quinquefasciatus that prefer these warmer temperatures? Is it creating a place for them to overwinter and breed somewhere warmer? On the left here, we also saw lots of like black widow spiders and stuff like that hanging out around. And then at the top on the left is an example of those egg rafts. We found quite a few of those while dipping in these catch basins. Oops. Oops, oops. So here's an overview of the methods that I was doing. So first, like I just showed, was collecting those larvae from the catch basins. Next was identifying larvae to their correct species, then extracting the DNA. Next was using dd-rad sequencing, specifically for that CQM1 resistance gene, as well as an ACE2 gene that determines Culex pipiens complex identity. So figuring out whether these are Culex pipiens, Culex quinquefasciatus, or potentially Culex molestus, which has never been shown in Utah, but it has been shown in California. And then lastly, using that dd-rad sequencing, we'll be doing population genetics analysis to determine gene flow, migration, kinship estimates, so determining the relatedness between these mosquitoes throughout the Salt Lake Valley. So it all starts with these catch basins, collecting the larvae, so using those dippers to get really deep. Some of them were dry, and then you'd have to like run to a nearby catch basin to try and figure out. It's really dependent on season and weather, so how much rain we've gotten. And so sometimes like you would have an original location in mind, and we'd end up having to go a few streets over or something like that just to find water and to find larvae within the water. Sometimes these were treated recently, and so that would impact. And so it was really just kind of being flexible and trying to make the study design work within the city. So we would dip down and then grab some larvae from the dippers, then identifying these larvae to species. So you would have to look specifically for Culex larvae. You're looking at the siphon on the bottom, and you're looking at what's called like siphonal tufts and pectins. And so it's really in detail. It definitely like took me a little while to get the hang of what I was looking for, but it ended up being pretty fun. And I actually found recently up at the top here on the top left is what I think might be like a nematode. I'm not honestly sure, but it looks very like monstrous and cool. So that was a lot of fun. And here are some close-ups just to show like how in detail you really have to look. So these siphonal tufts, and then these here are what's called pectins, these tiny little scratches right there. So next up was DNA extraction. So this is the step that I'm currently on right now. And so this takes a little bit of time because right now we're doing it with pestles. So each larva we're using individually to be able to track exactly like I said, the kinship estimates to see where it's coming from. And so we're using them in individual tubes and grinding that tissue up and then following just regular DNA extraction protocols from QIAGEN kits. So the ddRAD sequencing is the next step in our process. And we're doing this because it really allows for, ddRAD stands for double digest restriction associated DNA. And so it's sort of using restriction enzymes to cut at different sites throughout the genome. And this will be really good while we're doing these population genetics methods. And so that is the next step. And then the population genetics modeling is the last step in this process. We're set up to do that in the summer, and we'll be doing both like a large scale and fine scale analysis to make these kinship analysis and migration estimates to really determine like how far these mosquitoes are dispersing throughout the city, as well as where these genes are within the city. So are they moving in a certain pattern? Is this resistance spreading in some way that can be predicted? And is it within different treatment areas of that above ground and below ground habitats, as well as between Salt Lake City Mosquito Abatement District and the district south of them, the Salt Lake Valley. So we really want to just determine both fine scale, like within a city area, city few blocks, as well as between these cities, what's going on, and be able to correlate that to that previous publication a few years ago and be able to see really what's going on with these populations. So just some preliminary results because we are still in the middle of this process, and we're hoping to get more results this summer. The first interesting thing that we found the past few months is that we've actually been finding more Culex quinquefasciatus than previously believed in Salt Lake City. So up at the top here is a gel, and there's two different bands that we're determining for Culex pipiens and Culex quinquefasciatus. So the ones that have two bands would be what we would call hybrids, whereas the ones with just one band would be more likely Culex quinquefasciatus. So that was an interesting finding. And then we've also just in a preliminary analysis found that CQM1 resistance gene, which present throughout Salt Lake City populations. So that's interesting to note, and that is a good sign for our project that we're looking at a gene that is correlated with this Laciniobacillus sphaericus resistance and should be having some effect in our populations here in Salt Lake. And finally, just sort of like a current timeline of where I'm at. I'm currently doing the DNA extractions, like I said. This summer, we'll be doing the ddRAD sequencing and the population genetics analysis. And then hopefully this fall, I'll have some results and begin writing up a manuscript, depending on what we find, obviously. So thank you to everyone involved, everyone at the Sarman Lab, everyone at Salt Lake City Mosquito Abatement District has been such a big help getting this project going and getting me out in the field and working with everybody, as well as the South Salt Lake Valley people, and obviously PacVec and the CDC for funding this project. So thank you everyone for listening as well.
Okay, great. Thank you very much, Emily. That was wonderful.
Thank you.
Yeah, does anybody have any questions? You can either unmute yourself or put them into the chat. I was curious if you found besides the syrphid larvae, if there are other sort of insect or other sort of ecological observations you made in some of like if you made any correlations between like places where you expected mosquitoes but didn't find any, or vice versa, where you found lots of mosquitoes and if there were like associated arthropods there.
Yeah, I definitely did notice that like it was really climate based. Like after like a rain, we would go out and find a bunch, and then it'd be like I tried to go out, I think in like October of 22 or something, and like there was like nothing because it was all dry by then. So that was something that I think I hadn't really had in mind at first, and then realized that I had to be a bit more flexible with climate. But yeah, like ecologically, there was a lot going on in there, which was interesting. And then I think too, like a lot of people assume that it's like sewage and like really dirty water, but it actually isn't. It's just like organic matter from leaves and stuff. And so it really does create like the perfect breeding ground for mosquitoes, but then also a lot of other organisms end up liking it a lot. So yeah, it was kind of interesting to figure that out.
Yeah, it's something I've always been curious about, if there's other creatures at
Yeah.
level or other insects that like make for good breeding sites and other ones that maybe make for bad ones that
Oh yeah.
potentially affect ecological dynamics. Any other questions? Right. Well, if we don't have any more questions, I think we'll wrap this session up. And please tune in for our last seminar, which was going to be June 18th. And we shall see you all then. And thank you to Max and Emily for great presentations.